Okay, this lecture is covering osmosis, tonicity, and water movement. So here are the outcomes we will cover. And let's start by talking about gradients. So cells maintain gradients. So let's first talk about what is a gradient. For our purposes, a gradient means there's a difference in concentration. Another word for concentration is amount. So there's a difference in concentration on either side of the membrane. So this isn't a true gradient like we think of things going lots to little and everything in between. But there is a difference, and as molecules move, it does make this gradient. So the key is that there's a difference. We'll talk in a second about why this is important. There's two different types of gradients. There's a chemical gradient, and like this says, the concentration of a solute is higher on one side of the membrane than the other. So solute means stuff. Solvent in biology is water. So solute is dissolved in the solvent. So stuff. This could be ions. This could be sugars. This could be amino acids. Any kind of things that are dissolved in the water. Okay. So here they're showing you glucose. Lots of glucose on the outside, less on the inside. So there's a concentration or a chemical gradient because we're talking about a solute. The next picture over talks about electrochemical gradient. So that means there's an electrical gradient and a chemical. So electrical means charge difference. Or an electrical gradient means there's a difference in charge. So that means pluses or minuses. So think ions. So what that's showing you here is here's a whole bunch of different ions that have different concentration gradients. So there's less chlorine out here than inside. There's more sodium outside than inside. And it looks to be like less potassium outside than inside. So because these are charged, we call this an electrochemical gradient. They have a charge, and it is the solute. So why do cells have gradients? Well, cells use gradients as driving forces. I'll just write that. Cells use gradients as driving forces. Okay. So you can have a chemical driving force, you can have an electrical, or you can have an electrochemical driving force. One example that we've looked at is secondary active transport, where a cell will create a chemical or electrochemical gradient, and then that drives a second molecule in. This is an important concept in cell bio and physiology, so I just want to introduce it here. What we're most concerned about is the movement of water. Oops, I went the wrong way. So let's talk about the concept of tonicity. So tonic is going to represent the solute. Okay, so the amount of stuff dissolved in water. Tonicity is all relative to the side of the membrane you're looking at. And let me explain this. I should say, are comparing one side to another. Okay. So we're going to have three types of tonicity. Isotonic means same amount of solute on both sides of the membrane. Okay. So isotonic, same amount of solute on both sides of the membrane. Hypertonic means there's more solute on one side than the other. And hypotonic means less. Hypo means less. But it all depends on which way you're talking. So if you read these descriptions, it says the solute concentration outside the cell is hypotonic to the inside. So there's less solute outside of the cell compared to the inside. What I want you to note is if you looked at this the other way, the cell is hypertonic compared to the outside, right? More stuff inside than out. So you always need to talk about which side you're looking on relative to the other side. So let's look at these again. Isotonic. So in these diagrams, the big black excuse me, circle is the solute. And for these black circles, there's the same number on the inside as the outside. What I like to use better is actual values. So what's trying to be shown here is, say we have, we have a cell or something inside some liquid. Okay. Inside here you have 10% salt. Outside in the liquid you have 10% salt. Salt is the solute, and so you have equal amounts on the inside and the outside. It's isotonic. Hypotonic. Okay. So now there's less solute outside the cell than inside. The black dots are supposed to be water, I think. Let's look at a value. Okay. So inside our cell is 10% salt. Outside our cell is 2% salt. So the solution is hypotonic outside of the cell. Okay. So there's less stuff dissolved in the water outside of the cell. Hypertonic, just think of someone who's hyper, has like too much energy. This means more. And again, if we look at our solution outside of the cell, it's 30% salt. Inside the cell is 10% salt. So the solution outside of the cell is hypertonic, more stuff, compared to, oops, let me write hypertonic outside of the cell compared to the inside of the cell. So you need to be able to determine if a solution is hypertonic or hypotonic or isotonic given certain values. And you need to be able to, I don't know what you need to be able to do. I just lost my train of thought. Let's keep going. Whether or not a solute crosses the membrane, right, whether or not these big dots can move back and forth depends on the type of transporters a cell membrane has. That's our selective permeability. We're most interested in the movement of water because that can really affect a cell's health. So osmosis is the movement of water across the membrane. Water always moves from high to low concentration. And I should say from high to low water concentration. So this is just the same as diffusion. But because it's water and water's so important, it gets its own term, osmosis. So osmosis is the movement of water from high water to low water concentration. One thing I want you to notice is that the concentration of water is always the opposite of the concentration of solute. So if our blue is water and our red is sugar or solute, you could say that this side is hypotonic to this side. Right? There's less solute, less red dots on this side of the membrane compared to this side of the membrane. If there's less solute, there's going to be more water. So just imagine you have a set amount. You can have a mixture of solute and water, but I always think of it as equaling 100% or equaling, say, 10 molecules or something like that. So if you have lots of water, you only have room for a little bit of solute. Over here, it's just the opposite. If you have lots of solute, you only have a little bit of room for water. Okay? Water is always going to move from high water concentration to low water concentration. So let's take a look at our examples again. So we have our 3 beakers with different solution concentrations. Our cells are remaining constant with 10% salt. All right, here is an isotonic solution. That means the concentration of solutes in the solution is the same as the concentration inside the cell. Water will move back and forth across the membrane, but there is no overall or net change in the water concentration. Because this is an isotonic solution. So remember, in biology, nothing ever stands still. So water's going to move this way and water's going to move that way. And one way you can make sure you're correct is I like to think of these again as 100%. So if there's 10% salt, that means there's 90% water. And inside the cell, if there's 10% salt, there's also 90% water. So water can move out and water can move in. Water is in kind of equilibrium. And there's no gradient, right? It's the same. So the direction of water is going to be back and forth. Compared to this example, so we have 2% salt versus 10% salt. So this is a hypotonic solution compared to the water. I'm sorry, compared to the cell. So which way will water move? Well, let's figure out where water is at the highest concentration. So we have 98% water since 2% is salt. Inside the cell, these cells are all the same, we have 90% water. Okay, water is going to move from high to low, so water will move into the cell until it reaches an equilibrium. But we're just going to take a snapshot and say which way will water initially move. So in an isotonic, water will move back and forth, no overall movement. In a hypotonic solution, water is going to move into the cell, but you don't have to memorize this. You can always figure this out, and I encourage you to draw yourself a little diagram and figure it out, given a story problem. I try to do as little of memorization as possible because I always start to second guess myself. So if you fill in these numbers, you can see that water is going to move inside the cell. So how about this solution? More solute on the outside compared to the inside, so this is hypertonic. 30% salt, 70% water. 10% salt, 90% water. Water will move out of the cell. Now, some textbooks, excuse me, sorry, some textbooks state that water moves from low to high solute concentration. And this is correct, right? Water is moving from low solute concentration towards high solute concentration. And again here, water is moving from low solute concentration to high solute concentration, which means that water is moving from high to low water concentration. So I want you to see how these are just the reciprocal of each other. I don't like this, it confuses me, so I always just remember water is going to go from high water to low water. All right, try this one. Now I've given you something called molarity, which we haven't talked about, but what you can do is compare the numbers. And so a simple way to think about this is if you compare, this has high solute compared to the cell, right? 0.7 is greater than 0.5. If you have high solute, you have low water. So which way is the water going to move? From high to low. Okay, low solute, higher water. this would be a what kind of solution? Think about it. Hopefully you thought hypertonic. More stuff outside of the cell. Okay, take a look at the second one. Okay, you have less stuff outside, which means you must have more water. See how they're always reciprocal? More water is going to move to less water. So you're always comparing what's on either side of the membrane to determine which way water is going to move. Osmosis, which is the movement of water, can happen in two ways. We already talked about how water can go right through a membrane by simple diffusion. Sorry, I got the back to school cold. Okay, this is a slow process, but it happens. This is like passive diffusion, high to low concentration, though remember it's really passive osmosis. Now, many types of cells have a protein channel, right, like a little tunnel, called an aquaporin. It's selective to water, but water can move much faster through this aquaporin, and it goes in either direction. Both are going high to low. This is like facilitated diffusion, but we use diffusion to talk about solutes, so it's kind of facilitated osmosis. But I want you to know it's always going from high water to low water for both types. Many cells have aquaporins because water is so important, which we're going to see in the next slide. Cells that do a lot of water exchange, like cells in the kidney, have lots of aquaporin, or these protein channels. So why do we care so much about osmosis in cells? Is because different cells like different types of solutions. Animal cells prefer isotonic solutions. This is where they're most happy. You got equivalent amounts of water moving back and forth. If you're in a hypotonic solution and lots of water is coming in, the cell can lyse or burst. Because our cells, animal cells, only have a fragile cell membrane. If you put an animal cell in a hypertonic solution, water will leave and the cell will shrivel up, right? Because there's lots of solute out here, less water, so water leaves the cell. That's not healthy either. So animal cells prefer isotonic solutions. On the flip side, plants are a little bit different. Plants are most happy in a hypotonic solution. Plants have a nice cell wall. Okay. So they want extra water to come in, and remember they have a this little guy, central vacuole, to hold this water, and they have a cell wall that can resist the water pressure, and we call this state turgid, or we call it turgor, and this makes your plant stand up. Okay, So plants need to be overfilled. They prefer hypotonic solutions. If a plant is in an isotonic solution, it actually gets flaccid or starts to wilt, right? So if you've ever had a garden or a plant indoors and it gets a little wilty, you give it some water, and in a short time, it perks back up. If you've ever had wilty celery, you've left too long in the fridge, you can actually cut that celery, stick it in a glass of water, and it will go back to its state of turgor and crisp right back up. Now, if a cell has lost too much water, right? So too much water's gone out, and this can be, because of solutes, this could be dehydration. You're in a state of plasmolysis, or plasmolysed, but I say plasmolysis, which means the plasma membrane has pulled away from the cell wall. This is going to result in cell death. Okay, so if you've let your celery get too dehydrated, you can put it in water all you want, but it's not, excuse me, it's not going to recover because the cells are starting to die. Okay, so I want you to know where each type of cell is happy and what kind of solution and why. Okay, so let's make sure we went through all these outcomes. Discriminate between a chemical gradient and electrochemical gradient. We did that, both based on differences of concentration across the membrane. Chemical is just your solutes. Electrochemical includes ions or charges. We've worked on isotonic, hypertonic, hypotonic, so make sure you can define. Remember, this is based on the stuff in solution, these terms. So the solute, not based on the water concentration. So its name is it describes how much stuff is in solution compared from one side of the membrane to another. Okay, I should write osmosis. Osmosis can occur in two ways, simple or facilitated. Passive transport does not require energy, moves from high water to low water. We've done some examples of how solute concentration affects water movement, right? High solute, less water, low solute, more water. You figure out which way the water is going to move, and you use that to determine the direction of movement of water across a selectively permeable membrane if given solute concentrations, right? So you've done that. We'll practice this some more in class, but that covers those concepts.